Hello, my name is Dr. Rima Rachid. I am the co-director of the Food Allergy Program at Boston Children's Hospital. I'm an associate professor of pediatrics at Harvard Medical School. And I'm really happy to be here today to talk to you about the very important trial and collaboration we are doing with Dr. Alex Kurtz from the University of Minnesota Microbiota Therapeutic Program. I'm going to share my slides. So there is significant evidence that the microbiome plays a major role in food allergy. To give you some idea of the background, food allergy affects up to 11% of adults and 6% of children. And the prevalence has been increasing. There has been at least 18% increase between 1997 and 2007. And 90% of food allergies are secondary to the major 9 food allergens, which are milk, egg, soy, wheat, peanut, tree nut, fish, shellfish, and more recently, sesame. So food allergies could manifest by simple cutaneous lesions, skin lesions such as hives, but they often can be much more significant and concerning. Patients can have tongue swelling or even throat swelling with involvement of the airways, and even this could lead to the need to intubate a patient or to provide cardiovascular support because of anaphylactic shock. So in terms of natural history, while 80 to 85% of milk, soy, egg, wheat allergies remit by teenage years, that's not the case for peanut and tree nut allergies. They are usually lifelong, and we think that around 20% only might lose their allergies over time. Food allergies account for 200,000 ER visits per year, and there is an estimate of 150 to 200 deaths per year, predominantly from peanut and tree nut anaphylaxis. So the problem is that patients might react to very low doses. These are traces that they might not see in a food, either because of accidental ingestions or because of contamination of that food during the production or during cooking. And this leads to significant social limitations in play dates, in birthday parties and restaurants, especially in the setting of multiple food allergies. Patients often sit separately at school at the nut-free table, and this, therefore, will generate significant anxiety for patients and their parents from developing an allergic reaction. The standard of care for most of the food allergies is to just avoid the food allergens and keeping rescue medications available for emergency use. There is one FDA-approved therapy for peanut allergy, which is oral immunotherapy. Unfortunately, this is a very tedious therapy that, if lifelong, not curative, does not work well in older patients or adults, older children or adults. So there is accumulating evidence that the gut microbiome plays a major role in the development of food allergy, and clearly we need to do a better job trying to find a way to treat food allergy or even to prevent it if possible. Now, this evidence comes from initially observational studies comparing the microbiome of healthy babies with food allergies, as we and others have done, functional analysis in the lab, but now we are at the stage where we are doing interventional trials. So this is an example of what we did in the lab. This is in Talal Shatila's lab. We took the stools of healthy babies and the stools of food allergic babies, and we put them separately into a highly allergic mouse model that can have very severe anaphylaxis. Anaphylaxis is measured in the mouse by a drop in body temperature because it can lead to vasodilatation, which is dilatation of the blood vessels because of the allergic response. The mouse is then sensitized, and then we, by giving small doses of egg protein, and then we challenge the mice by giving large dose of egg protein. And what you see here are the mice that did not receive any fecal transplant. There was significant anaphylaxis and drop in body temperature. This was even further down in mice that received the fecal transplant from food allergic babies, while in the mice that received the fecal transplant from controlled babies that did not have food allergies, there was protection from anaphylaxis. So that, of course, was very, very appealing to us. Why? Because around that time, fecal transplantation in the form of encapsulated forms came in, and it was found to be very efficacious in treating C. difficile colitis. So we thought, wow, that would be a great way to see whether our patients, our food allergic patients, might actually respond to that therapy. So we launched a phase 1 open label trial to evaluate the safety and efficacy of fecal microbiota transplantation in peanut allergic patients. So as you know, fecal transplant can be administered via colonoscopy, via enema, via tubes that goes into the stomach or the intestine, but the capsules were very attractive to us because they appeared really non-invasive for patients who have primarily a food allergy. We got the fecal material from open biome. We selected donors that had no food allergies. The caveat here was that we had to train the donors to avoid peanut and tree nut because we wanted to avoid the possibility of our patients reacting to some food that could still be present in the stools if the donor have eaten either peanut or tree nuts. So we trained the donors on avoiding peanut and tree nuts even in trace amount, and we tested the stools for the presence of traces of peanut and tree nut at LCMS Limited in North Carolina. So the objective of the study was first to see whether it is safe and tolerable in the young adults with peanut allergy. But we also decided to have secondary objectives of efficacy. We wanted to see whether the threshold of reactivity to peanut could go from 100 milligram peanut protein or less. This is less than half a peanut. And we start food challenges at 1 milligram. So some patients will react to 3 or to 10 milligram. And we wanted to see whether we go up to 300 milligram, which is about a peanut and a half at 1 month and 4 months post FMT. We even wanted to see whether we could go up to 600 milligram, which is about 2 to 3 peanut at 1 month and 4 months post FMT, and to evaluate the changes over a year time. You might wonder why 1 month and 4 months? Well, it's simply because it was just, let's call it an educated guess. We really didn't know if the immune response was going to respond. When will it respond? So we enrolled initially 10 adults, age 18 to 34 with peanut allergy. Some of them had also a concomitant tree nut allergy. They came in for screening. They did their food challenge. Then they did the administration of oral fecal microbiota transplantation. These are the doses they received within 3 hours. You see in total, there are 36 capsules. That's the dose given for C. difficile infection patients. And then they came in 1 month later and 4 months later to do food challenges. In terms of safety, there was no serious adverse events, no allergic reaction, just mild adverse events that lasted a few hours to 2 days. And in terms of efficacy, we had 3 patients out of 10 that actually achieved efficacy. 1 patient went from 100 milligram to 300 milligram reactivity dose at 1 month, and that persisted at 4 months. 1 patient reacted at 10 milligram peanut protein, went up to 300 at 1 month and 600 even at 4 months. And 1 patient reacted at 100 milligram and tolerated all doses. That's a cumulative of 1,043 milligram of peanut protein, which is around 5 peanuts, and again, tolerated all doses at 4 months post-FMT. So that was very promising. So we wondered what would happen if we were to administer FMT, but this time with antibiotic pretreatment. Would an antibiotic pretreatment create a niche in the gut for FMT to implant better? So we did exactly the same procedure with and administration in these patients, except that we had them take oral vancomycin, neomycin, and metronidazole for 4 days, followed by a 2 days hiatus for the metronidazole to clear up from the system. And then again, we did FMT, and then we repeated the food challenges. And here again, we had no serious adverse events, no allergic reactions related to FMT. But in terms of efficacy, we had 3 out of 5 patients who responded. Patient 22 reacted at 100 milligram. These 2 patients could not do their 1-month food challenge because of the COVID pandemic, but they both actually went up to 600 milligram at 4 months post-FMT, and patient number 27 reacted to 30 milligram, went up to 600 at 1 month and 4 months post-FMT. So the outcome summary is that the study was safe. Without antibiotics, there was 30% efficacy just from 1 FMT. And with antibiotics, there was 60% efficacy response just from one FMT. I'd like to let the audience know that it takes about 6 months of oral immunotherapy to reach that level of peanut tolerance, and that oral immunotherapy is very challenging in adults. So adults per se are very difficult to treat. This efficacy effect was prolonged and persistent till at least 4 months post-FMT. I will only go very briefly over the mechanistic studies. Here again, what happened is that we had a fecal transplant going from a healthy donor into a food allergic patients. We had 6 patients that were responders and 9 that were non-responders. So we took the stools of the responders before they were transplanted and after they were transplanted, and we put them into a highly allergic mouse model, and we did the same with the non-responders. You see here that when the highly allergic mouse model received the stools of the responders at baseline before their transplant with the healthy microbiome, there was no protection from anaphylaxis. There was a significant drop in body temperature. But when we actually took their stools after they were transplanted with the healthy microbiome, there was protection. There was a very nice protection from anaphylaxis. This wasn't observed in the non-responder stool, whether before or after FMT. And the immunological changes in the mice also were very nice, as their total IgE went down, their ova-specific IgE went down as well. So with these promising results, we are partnering now with Dr. Alex Koretz at the University of Minnesota Microbiota Therapeutic Program to launch a phase 2 double-blind randomized placebo-controlled trial evaluating microbiota transplantation therapy in children with peanut allergy. So as you know, MTT is a form of FMT that is highly concentrated in bacteria. It contains less than 1% fecal material. It is currently being evaluated in autism, inflammatory bowel disease, and cancer patients. And what is nice about it is that, number one, the dose that is given to treat C. diff is only 5 capsules. So this is really nice. We're going down from 36 capsules to 5 capsules. That's definitely very attractive in terms of practicality, especially in pediatric patients, but also the fact that it doesn't have to stay at minus 80 degrees in order for it to be administered to patients within 90 minutes of thawing. It actually can stay in the fridge at 4 degrees Celsius, which is around 39.2 Fahrenheit for 6 months. And that becomes also a very attractive option for home administration. So now we are able to evaluate whether prolonged administrations of MTT can actually treat food allergies, and if so, how high can we go with the threshold of reactivity in pediatric patients? So this is the design of the trial. This is a trial that they were going to do in peanut allergic patients aged 12 to 17. They're going to come for screening. They will undergo a food challenge. Then they will be randomized on a 1-to-1 ratio, either to receive vancomycin and neomycin, followed by 5 capsules of MTT. The antibiotics will be given for 7 days, then MTT 5 capsules, or placebo, followed by placebo. And then they will go home, and those who received MTT will receive 2 capsules of MTT over 27 days, while those who received placebo will continue on receiving placebo for 27 days. And then we will repeat the food challenge at 1 month post-FMT, 4 months post-FMT or MTT, in order to see how different is the threshold of reactivity in these patients. We are aiming for 24 patients, and as of August 7, we have, which is the day I am actually recording this, 2023, we have had screened 2 patients. So finally, I would like to acknowledge all the trial participants, my collaborators, Talal Shatila and his team, Emmanuel Stefan Victors, who did the mouse studies, the clinical research team and co-investigator, my collaborator at MIT, Eric Alm, Libby Homan at MGH, Robert Voisker at LCMS Limited, and Open Biome. This is our source of funding. I'd like also to thank Alex and his team, the Minnesota Microbiota Therapeutic Program, and of course, ACT. I really would like to thank you very much, because it is so important to support these type of trials, because these type of trials are truly pushing the envelopes, trying to find better ways to treat our patients. Currently, the standard of care is extremely suboptimal, and we really would like to try to find a way to better treat food allergies, better treat and potentially even prevent and cure food allergies. What you are doing is really critical, because you are helping changing the landscape. You are truly giving hope to patients by supporting these type of trials, by supporting these type of programs that are truly critical in understanding the microbiome and the diseases that are affected by the microbiome. In fact, by doing this study, we will not just learn about food allergies, we will learn about other diseases, how the MTT will implant, what will it mean in this particular type of patients? How can we translate that to other diseases? How long do we need to treat these patients? So I cannot thank you enough for all the support you are providing. Thanks again.